nucleic acid-based analytic methods for microbiology. Some advantages of molecular methods is it is rapid, so we get faster results compared to culture-based methods. It is specific, so it's highly specific identification at the species or even down to the strain level. It is very sensitive, so it can detect low numbers of organisms even if they are non-viable. It can do direct detection, so it can detect pathogens directly from clinical specimens. There is no need for viability, like mentioned earlier. That is useful for organisms that are difficult or slow to culture. There is high throughput, so it can test for multiple targets simultaneously. Some disadvantages of molecular methods, though, is the cost. It is typically more expensive due to reagents and equipment. Technical expertise, so it requires trained personnel and specialized equipment. There is limited scope, so it usually targets specific pathogens, so it may miss unknown or unexpected organisms. We may also get some false positives, so possible contamination or detection of non-viable DNA. And no antimicrobial susceptibility data, so it cannot determine antibiotic resistance profiles without additional testing. It also has complex interpretation, so results sometimes require clinical correlation and expert interpretation. So molecular methods. With the launch of molecular methods, there's actually a variety of different techniques. We have hybridization, amplification, and the sequencing. In hybridization method, it's based on a base sequence homology or relatedness of two strands of nucleic acid. So nucleic acid strands will complementary base sequence that bond specifically and form a double-stranded molecule, which is a duplex or a hybrid. Amplification, this basically does what it says. It multiplies the target nucleic acid without having to cultivate the organism's sequencing. Is an enzymatic digestion of nucleic acid, which determines the organism's blueprint. In hybridization of the assay requires detection or identification of two nucleic acid strands of one strand, since we have the known, otherwise known as the probe, and the other strand, which is the unknown, which is our target. So the unknown is going to be from the patient. There are four basic steps. They are, we have a production and labeling of a single-stranded probe nucleic acid, preparation of that single-stranded target nucleic acid, mixture of a hybridization of a target in the probes, and then detection of the hybridization. Detection is done by a reporter molecule that binds or complexes with the probe DNA after hybridization. The reporter molecules can be radioactive, which emits radiation detected by imaging equipment, chemiluminescent, which produces light detectable by special cameras or sensors, or other commonly used tags can include biotin avidin system. So biotin is attached to the probe, which binds strongly to avidin, conjugated to an enzyme or marker. Digoxin is a plant steroid label that can be detected using anti-digoxin antibodies conjugated with enzymes or fluorescent tags. For PNA, or peptide nucleic acid, so synthetic DNA analogs with a peptide backbone instead of sugar phosphate, or FISH, which is fluorescent in situ hybridization. This is a technique using fluorescent probes to hybridize specific nucleic acid sequences inside cells. So PNA FISH targets species-specific rRNA sequences of microorganisms. The PNA probe penetrates the organism's cell wall and hybridizes to the rRNA inside the organism. This results in fluorescently labeled organisms visible under a fluorescence microscope. Can also be used directly on positive blood cultures to rapidly identify pathogens, and there are FDA-approved kits for Staphylococcus aureus, Candida albicans, in differentiating between Enterococcus faecalis from other Enterococcus species in blood cultures. This method speeds up pathogen identification significantly compared to traditional culture methods. With an amplification method, otherwise known as polymerase chain reaction or PCR, you're going to hear a lot about this as it is a very influential test and it was used throughout the pandemic. This utilizes a process of nucleic acid replication that makes many copies of the target DNA. So there are three steps. We have an extraction and denaturation, we have annealing of the primers, and then primer extension. So the extraction denaturation phase, we release or extract the nucleic acid target from the organism using either heat, chemicals, or an enzyme using what we call a thermocycler.
The target nucleic acid is then denatured to a single strand, which occurs at 94 degrees Celsius. Primers are then used to anneal or hybridize to the nucleic acid target.
Primers are short DNA sequences that provide a starting point for DNA polymerase to synthesize new DNA strands. Taq polymerase, a heat-stable enzyme from Thermus aquaticus, is commonly used in PCR because it withstands high temperatures during denaturization. Each cycle produces 2 double-stranded DNA fragments from each target sequence. The cycle is repeated 30 to 40 times, leading to the production of approximately 10 million copies of the target DNA. These DNA copies are called amplicons. The amount of amplicons is measured using a labeled probe, which allows detection of the target DNA. If you're in my class, there is a video on this slide to watch, so make sure you download the PowerPoint. Then we have real-time PCR on an automated instrument. So by definition, these are instruments that combine nucleic acid amplification with measurement of amplified product in real time. Some advantages is it combines thermal cycling amplification with fluorescent detection of amplicons in real time. Amplification and detection occur in a single vessel, reducing cross-contamination risk. Quantitation of product amount is possible, allowing measurement of the original target amount. The procedure time is significantly reduced compared to traditional PCR. So the difference between PCR and real-time PCR is PCR amplifies specific DNA sequences and is usually analyzed after the process, giving a yes or no result. Whereas real-time PCR amplifies DNA and measures it during the reaction using fluorescent markers, allowing for faster results and quantification of the original DNA amount. So real-time PCR is more sensitive and reduces contamination risk since detection happens in the same closed system. Once again, there's a video down below if you're in my class, so make sure you watch that. The nucleotide sequence of a genome serves as the blueprint for an organism. Identifying an organism is possible by determining its DNA sequence. PCR combined with sequencing is especially useful for identifying mycobacteria and nocardia species. Enzymatic digestion breaks DNA into fragments. These fragments are separated using gel electrophoresis. Stained bands in electrophoresis create unique restriction patterns for different DNA regions. Differences in restriction patterns are called restriction fragment length polymorphisms. Restriction fragment length polymorphisms reflect variations in nucleotide sequences and are used to identify the organism. And we'll talk about each one of these next.
So with direct detection, we have a nucleic acid hybridization and a target or a probe amplification method that's most commonly used for direct detection in clinical specimens. Specificity, both methods are driven by specificity of the nucleotide sequence, so a positive assay indicates presence of an organism and identification of that organism, so your most direct method for organisms that don't require antimicrobial susceptibility. So for example, we typically don't do antimicrobial susceptibility testing for group A strep, so we can ID it, but we don't need the antimicrobial susceptibility testing. So therefore, we don't need it, or it isn't growing on the plate, and our limitation to this, it's only good for 1 or 2 organisms per specimen. You get some individuals, like the wound cultures, will have multiple organisms that have to be worked through to identification, so each specific. Sensitivity allows a not sufficient amount of organism or if the specimen contains interfering substances. So sensitivity is enhanced by those amplification methods. We're able to detect 9 non-viable organisms, and you're also able to detect and ID organisms that don't grow well. It can detect unknown agents and has the ability to quantitate infectious agents from the specimen. So examples would be HIV, CMV, which is cytomegalovirus, hepatitis B, and hepatitis C. So again, you can also take an organism that's grown in culture and then apply one of the nucleic acid-based methods. So hybridization and amplification, or RFLP, can be used to identify organisms and culture criteria to determine using molecular techniques, which is going to increase speed, accuracy, and cost. There are slow-growing organisms, so an example would be like Mycobacterium. It can take 6 to 8 weeks. Fungi can take 2 to 3 weeks or longer. So it takes that week just to grow them, and then you have to have an ID system that sometimes is not available for another 24 to 48 hours.
So molecular methods can also be used to determine the organism virulence. So it can detect the toxin genes in certain bacteria. It can also detect resistance to antimicrobial agents. So like an example would be a detection of the MEK-A gene in Staphylococcus aureus. That could indicate that it is methicillin resistance, which is called MRSA. And or it could do the relatedness to another strain of the same species. So it can do whole genome sequencing for detailed strain comparisons in epidemiology.